Let's just wait for my screen to share. Oh, yeah. So what I'm going to do is I'm going to go over kind of what our protocol is in our entire pipeline. So I'll probably skim over a lot of this just because it was mostly covered in the more technical aspects in the sessions yesterday. But the long and short of it is, like everyone was speaking to yesterday, our challenge is to deal with all these refractive index mismatches in tissue. And, the details of that, Casey went over yesterday, but the long and short of it is the way to deal with it is to remove the lipid membranes. So what we do at LifeCanvas is really provide solutions from start to finish, from the preservation step all the way to the image analysis steps. And what I'm going to discuss here is kind of the protocol outline that we do for each of these steps. So this is both in kind of the reagents and devices that we provide, as well as what we do ourselves in-house in our contract research services. So to recap, we both process tissue for researchers. So researchers send us tissue and we do it ourselves. And we provide protocols, equipment, and reagents to clear, label, image, and analyze tissue for users. So this is just a general overview of the process. It's based on technologies that Casey went into a bit more detail yesterday, but it's basically the shield and stochastic electron transport protocol on the tissue processing side, followed up by light sheet imaging and 3D image analysis. So the first kind of step that we do with tissue or in our protocol is shielding it. So Casey already went over kind of the advantages of shield. It's an epoxy process. So the epoxy group is polyfunctional, so it reacts with a lot of different functional groups like primary amines, carboxylic acids. So it provides a further degree of cross-linking compared to formaldehyde fixation alone. And that's kind of important because we're doing additional processing steps as well as to preserve the mechanical properties as well as the chemical properties. Compared to the older version of this kind of technology, Clarity, Shield is a lot easier to use. It's just an incubation versus like a hydrogel embedding process. So the actual steps is, after we do a standard PFA perfusion, you just dissect the sample out, typically a mouse brain, incubate in one solution for about what we call our shield off solution. So this shield off solution is what actually contains the epoxy chemicals. And that just lets the epoxy distribute throughout the brain. And the kind of buffer that it's in doesn't allow it to react quite yet. And then after it has a chance to penetrate through the sample, one would then incubate the sample in a shield on solution, which allows the cross-linking reactions to happen. So the shielding process is a simple, kind of two-step incubation process that happens for the shield off stage around 4 days, and then 1 day for the shield on step. Then after what we call shielding or tissue preservation, we move on to clearing. So there's a little bit of confusion around the term clearing. So, sometimes clearing is just like the delipidation part, or sometimes it's referred to as the whole, process up to refractive index matching. But kind of the point here is at this stage, we're just aiming to remove the lipid membranes. At the end of this stage, you won't actually get fully transparent tissue yet because the refractive index medium hasn't been matched yet, but it'll be kind of a translucent stage. So again, the technology behind this is the stochastic electrotransport technology that Casey went over yesterday. So long and short of it is that we're using a rotating electric field to drive SDS micelles in and out of the tissue to remove the lipid membranes. And compared to passive clearing, this electric field speeds up the process, but by having it rotate, we're not risking sample damage by pulling the sample too much in one direction and causing deformation. The basic steps to use this kind of device, which we call our Smart Clear device, is you take the sample that's been preserved, you put it in a little tissue biopsy bag, put it inside a little sample cup, fill the put the sample in the machine and put in the right buffers. So there's two clearing buffers, one that flows on one side of a set of nanoporous membranes and one that flows through the sample. And the reason for the separation between the buffers is largely just to preserve the buffer life by preventing too much detergent from being electrically oxidized, as well as protecting the sample from any like electrolysis products that come from the electrodes. Then at that point, you flip the machine on, all the pumps are running, you flip on the electric field, and you wait approximately 5 to 6 days for a whole mouse brain. And you end up with actually not something in the picture on the right here, that's after index matching, but kind of a translucent thing that resembles like a gummy bear almost in terms of like transparency. Yeah, and then so the reason why we have to index match well is because of course imaging comes down the line. So that kind of step comes later down the road. So after you're done clearing, if there's just endogenous reporters, you can actually go straight into incubating it in what we call our easy index solution. And this kind of this is just a refractive index matching solution that actually makes the tissue fully transparent after removing the lipid membranes. And this has just been developed to be optimal with our entire setup. And so you can see kind of in these pictures, index matching is still important after removing lipid membranes because any sort of refractive index mismatch, especially when you go deeper into the sample, can cause blurriness. But a lot of the time, we're not just interested in endogenous reporters. We also want to stain for things. So, some of the common targets that we've discussed yesterday, like CFOS, IBA1, GFAP, beta amyloid plaques or something. They're all of interest depending on your research question. And what we find that with a lot of clearing protocols is that once you clear the tissue, you still have to label it. And passive labeling is kind of the traditional way to do it. But there's two problems with passive labeling. One, as Casey mentioned yesterday, diffusion over millimeter, centimeter length scales of antibodies into tissue is extremely slow. So, as you scale up like thickness with you exponentially increase the labeling time. The other problem is that even if you can somehow magically make molecules move faster, antibodies will bind to the first kind of binding site that they have the opportunity to, right? So even if you could speed up diffusion, you get what you see on the left here, preferential binding to the outside of the sample versus on the inside. So for something like a brain, often we will want to look kind of below the cortex and into the internal areas. And so even if we can somehow speed up diffusion, we still have this issue that we have this kind of surface bias, which is often not ideal. So what you'll see with a lot of passive labeling is kind of like a ring of labeled, of like well-labeled tissue on the kind of the crust of the brain and the kind of like this undercooked core. So we deal with these two problems, again, using this electrotransport technology. And this essentially occurs through like a pH and detergent sweep. So we use these electrochemical reactions to change the binding affinity throughout the run driving antibodies in. So how that works is that as the electrochemical reactions go on, there's acidic byproducts that are produced, and so that changes the pH from conditions where antibodies don't like to bind, which is kind of in the 9.5 range, and then eventually brings down the pH to a more neutral pH where antibodies can actually bind. The other thing at play is that we use detergent that is also electrochemically oxidized that also inhibits antibody binding. So that way we can actually get uniform labeling throughout the tissue compared with passive. And the steps to actually do this is similar to what we do with clearing. There's a device that we use, and the steps are similar. You have a little sample cup. You put your sample into the cup. You add your antibodies. You turn on the device, and you let it run for about 14 hours for the primary labeling step, and then you wash it out. And then there's another, if you want to put a secondary in, there's another kind of a 12-hour step. And all this stuff is more detailed in our active protocol. In-house, we actually use a different device than the one you see here. This device can only take 2 samples at a time, essentially. But in-house, we actually use a device that can label or clear up to 6 samples at the same time, and is substantially smaller than the devices that you see here. They're not shown here because we haven't really released them officially yet, but if you have any questions about those, you can email me later. And then finally, when we actually have a labeled sample or has some endogenous reporters, it comes time to imaging. So we also provide an imaging solution, which the steps are largely similar in terms of protocol to any light sheet microscope. You would mount your sample in an agarose gel that is also index matched, and place it inside the microscope where it scans. So the first step is you have to index match your sample. So you incubate it in the index matching solution we call Easy Index. You set up the microscope. So all the settings that are usual with fluorescence microscopy, like where you're going to image, what channels you're going to image, and focus the image, and then we image. So the difference here that we found that's important actually is not all light sheets are the same. We kind of imagine like a light sheet as kind of a flat, perfectly planar sheet of light, and then you can image perfect kind of Z planes in your sample. The reality is that when a light sheet is formed, it's a Gaussian distribution. So the edges of a light sheet will be quote unquote thicker than the center. So if you take a image with a light sheet, you kind of have to keep in mind on most light sheet microscopes that the Z resolution will be better at the center of a tile rather than on the edges. We get around this by using axial sweeping technology. So what this does is that the center, we only take the image from the center of the light sheet, and we sync that up to the pixels on the camera. And then we move the pixels that we detect along with the center of the light sheet throughout the tile. So we're always kind of getting that center spot. And that helps, when we're starting to get into thicker and thicker samples, deeper and deeper into the sample, and illumination coming from both sides, that we kind of maintain the ideal axial resolution of the light sheet throughout the sample. And so typically in our CRO services, you can set up a light sheet experiment in 15 minutes to half an hour, focusing, loading in the samples, all that stuff. And then acquisition depends on the size of the sample and the magnification and how many channels. So a whole mouse brain at 3 channels takes like 3 or 4 hours if you want to acquire all 3 channels. Okay, and then this is not all just about pretty pictures. We actually want to get some data out of it. So we do have a software that we developed in-house that we use to conduct image analysis from these light sheet images. In short, by aligning them to the Allen Brain Atlas and picking out relative qualitative inputs. So we can do things like intensity by brain region, or cell counts by brain region, or depending on the markers. So typical things that we do are CFOS cell counts by brain region. We can do things like NUN, beta amyloid kind of plaque quantification, or just straight up fluorescence intensity measurements. So how this works practically is that there's some software that we have that automatically, semi-automatically aligns the light sheet 3D imaging data to the Allen Brain reference atlas. And then there's a manual kind of correction step because, samples can be quite variable depending on how they're handled, where they're from, to kind of fine tune the alignment by moving correspondence points. At that point, so this manual step typically takes around half an hour to an hour for a research tech to complete. And then at that point, once the alignment is good, we throw everything into cell detection or fluorescence intensity measure model. That runs on the computer, classifies the area or cells as either positive or negative, and then that's projected back onto the Allen Brain Atlas to give all the cell counts, all the intensities by brain region. So here's some of the videos of the detection in action. So this is PV throughout the whole brain, and in the red, this is the model picking out the cells. And then this is a similar thing going on with CFOS near the hippocampus. And the green is the immunolabeling we have with CFOS, and the red is the cell detection. And then similarly, this is just kind of an intensity mapping of Thy1 YFP. And on the left is the image, and on the right is kind of the segmentation of the intensities on the Allen Brain Atlas. So we have several user sites that have adopted some or all parts of this pipeline, and we just ended off with a pretty picture of a mouse intestine portion. And if you have any questions after this, my email is brian@lifecanvastech.com. Thank you.
Okay, thank you. Okay, I think it's time to start the next session of the workshop. So at this point, we're going to get a little bit more into how to image your cleared and expanded tissue. So we're being joined by Oliver and Pascal. They are our local Zeiss team based in Vancouver and been helping us with imaging for many years. And Damien Wheeler is also joining us from Translucence Biosystems. I guess our interactions with Damien started after an earlier meeting we had this year where the presenter was using technology from Translucence in combination with the Zeiss microscope to image very large tissue samples, which is of interest to many of us. So I think I'll invite Oliver to share his screen and start us off with a discussion about conventional light sheet microscopes. Oliver, are you there?
I am there. Good morning, everyone. And I hope you can hear me and see my screen. Is this correct? Can people nod or confirm?
I can hear you and I can see your beautiful slides. So
awesome. Okay.
Let's go ahead.
Well, thanks for inviting me. I listened on and off to some talks, and it's great to see kind of an interest in this, and I would say also the sharing of the knowledge and the ability to really now do microscopy in a way that we weren't even thinking about. Let's say 5 years ago, even maybe Jeff, 10 years, there was a lot of grant writing about mesoscale and large scale imaging and biology, but there weren't really any methods out there. So in a way, my talk is a little bit more humble, I would say, because all I tell you about is options how to, once your tissue is cleared and prepared and the biochemistries are correct and stained, etc., how to image this. There are different methods to do that, and I will really focus today on light sheet microscopy, and there's even different flavors of light sheet. So I'm going to break this up into two sections. One being the classical, I would say by now, a Gaussian light sheet system, which I show you here. I start out with this. There's a system like this on the UBC campus, and we have heard from other users around the country, and that this system is being used for a lot of the kind of large volume imaging. So let's start out with this. The Light Sheet 7, that it's kind of our brand name for this, maybe not very imaginative, but here you go. So this is our Gaussian light sheet system. And why do we need any kind of special system to look at large expanded or non-expanded large volumes of imaging? Well, first of all, what is a light sheet and why is a light sheet? The light sheet simply refers to the illumination modality in which we are creating a very thin or selective plane, also sometimes called SPIM for selective planar illumination, a selective plane of illumination that is orthogonal and a 90-degree angle to our detection objective optics. So this is called the horizontal microscope design. There are plans, the original plans for this are actually over 100 years old. So the idea is old, but the implementation of this kind of, at least in the commercial realm, is new. And so we now have a separated light path for the excitation, so illumination, and for the detection. So this uncoupling of this allows us to image something within the sample plane without coaxial illumination. I'll go into a little bit more further detail in a second. Another problem is if you want to image a very large object, let's just say here a whole fish, and this could be a cleared brain, it doesn't really matter. This fish is supposed to be still alive, as you can see in a second. In order to image to the whole fish, you have to get very close. And when you get very close, because of the working distance of objectives, you oftentimes bunch up, so to speak, against your specimen. So there's a natural or optical limitation to how thick you can actually image into a tissue with respect to the working distance and depth and the depth of field and focus of your objectives. So typically, you have for fluorescence experiments, you would have a high numerical aperture lens with a pretty short working distance. And you can see this here at this working distance, you have a field of view that is also exceedingly small. So this kind of optical setup doesn't lend itself for high volume imaging. Plus, you now concentrate through this high NA all your photons within that focal plane into your oftentimes living. We talk about clearing here and expansion, so not much stuff living there, but these instruments are originally also made for live organism imaging. So you concentrate all your photonic power and load into a small focal volume, which leads to sample heating and photo damage. So to avoid this, light microscopy, again, now instead of using a coaxial illumination like a wide field or confocal, in which the illumination, the excitation comes through the same objective lens that the detection is being done by, is now illuminating from the side. That's the number one trick. So we basically have this decoupled, we have an orthogonal angle. And that allows us to now, from the side, if we have a detection object that's large enough to read out a whole section of the illuminated object at the same time. So you're fast and you're selective in terms of illumination. So we have here similar in concept to a multiphoton in illumination procedure that is by default an optical sectioning procedure. So large field of view, optical sectioning on the fly, allowing for fast acquisition and much less photo damage because we don't illuminate all the fluorochromes above and below our actual scanning or detection plane. Another cartoon just showing this, instead of illuminating everything that is basically in the column of the objective front lens, regardless whether these fluorochromes are in focus or not being illuminated, wide field or confocal, we now only illuminate the ones that we actually image. And then you, for confocal, for example, you would scan your object along the detection kind of field of view. Again, exciting all fluorochromes throughout the z-axis, but with light sheet you only do a single plane illumination and detection. And that's of course also much faster. This is kind of the setup, how it looks in our system. There's a detection chamber in there, and Damien from Translucence will go into a little bit more detail about different models and ways to actually put your sample in there and accommodate for very large samples. But here you see you have a detection objective. This is a water dipping objective that actually interfaces with the aqueous media within this chamber, and you have two, not one, but two light sheet forming objectives. These are your excitation objectives, so to speak. And you can see here is we are forming a, let me see the Gaussian shape here, a Gaussian shaped light sheet that at its flattest, thinnest waist is exactly where your detection of your fluorochromes will happen. So again, orthogonal setup, and to create this light sheets, there are different methods. One versus kind of a standing static light sheet, and one that's via a scanned laser. And in our system, we basically do a combination of both. So this is how we create that Gaussian light sheet within that sample chamber. And you can see this here. So here, the detection optics, we're now looking to the front of the chamber, and you can see kind of the Gaussian shape, the waist of the beam for the standing light sheet versus the laser scanning light sheet. Okay. So this is how we form a light sheet, and this is how we detect the light. Then all we have to do is somehow, again, hang our specimen in there. It's ideal for living specimen, and it was really built with developmental biology in mind to a certain degree. I don't like to use the word, but it kind of got hijacked by neuroscience, as many kind of things do get, for better mostly, because people wanted to look at larger architecture and mesoscale imaging for in whole brains. And so clearing was really not in the beginning the main application, but we now have adapted light sheet optics, chamber optics, and protocols in place to deal with the refractive indexes needed for cleared and expanded specimens. Another really important feature is the freedom of positioning freely your specimen relative to your detection, which is not obviously working on a classical compound microscope, because your specimen is not 3D orientable on a cover glass or under a cover glass on a slide. So we can do this here. We can move this in X, Y, and Z and rotation. So it's really 4D positioning that we can do. If it's for living specimen, we can have an aqueous and fully temperature and CO2 controlled chamber here to keep things alive. As I showed you before, we have two objectives for illumination. So we can actually do what's called dual-sided illumination. That means we can scan from the left-hand side with the sheet coming from the left-hand side. And we can do the same for the right-hand side. And when we then on the fly basically paste or fuse these images together, we have created kind of an homogeneous illumination as possible, and having objects that are basically, got a kind of an isotropic illumination. The next thing to deal with thicker specimen and light sheet illumination is anything that's in the light path of the forming light sheet that is being brought into the specimen, and that absorbs light or blocks light will show shadows. And you can see these shadows then will represent kind of stripe artifacts. So we have something else built in, kind of a scanning element built into the system that brings actually the light, instead of just from a single point, it brings it in from multiple points. It's called pivot scanning. So I'm kind of in a very fast fashion rastering this light sheet in different angles back and forth. We can basically bring light in from the sides of the blockage, from the sides of the piece of the tissue that does not let light penetrate, and therefore achieve a homogeneous illumination. This clearly shows you that when you look at your specimen, here is zebrafish, and kind of this is mounted in agarose. This is the kind of the 3D rotation relatively to your objective, and this allows you to position your specimen at the right angle relatively to your detection objective. It also allows you this 4D rotation, or the kind of the rotation around its axis, to take images from different angles of your specimen and then fuse them together. In our software, this is called multi-view. And again, it's something that you cannot do on a compound microscope, but it allows you to get different views of your specimen and fuse them together in 2D, 3D, or 4D. The more technical side of it then kind of looks like this. These are our imaging chambers. The chambers are shown here relatively to specific light sheet forming objectives, optics, and detection optics. That could be either air, that means they don't interface directly with the chamber, or there could be even dipping lenses. There are some lenses that are specifically meant for the refractive index of typically clearing lenses. Now, I would say for most kind of mesoscale applications, you really go with a 5x or even 2.5x detection optics, because you need extremely large volume. Then this is coupled with a single or dual camera setup. Dual camera simply makes the whole thing faster, because you can excite 2 fluorochromes at the same time and image 2 fluorochromes at the same time with the right dichroic and emission filter setup. On the right-hand side are kind of the sundry little holders and all the kind of bolts and pieces that allow you to basically lower your specimen into the chamber in various ways. And there's some magnetic setups. There's some stuff that requires gluing. There's something that you can basically put it on a little plate. And again, this is just simply for bringing your cleared specimen into the chamber and keeping it there stably. Last thing I want to show you, and you have to kind of bear with me on this one, is I want to bring up this little introduction video, and I hope it's going to start up for me and for you. And you have to tell me whether or not you see this, but because I might have to change the way I share my screen. So if anybody wants to comment via audio, whether they can see this or not.
I don't see a video.
Okay,
I
so
see
let me stop my
slides.
share quickly, or I'll go new share, and I'll do this, I guess this, and I will share this. So this just shows you, kind of in a 3D fly through how now the kind of the system is built up, built optically speaking, and how these things come together. So this is the front of the instrument, the chambers, and there you have a front lens camera with an LED transmitted light that gives you an overview image that lets you position your specimen within the chamber. Those are the focusable illumination optics, light sheet optics, and these are the detection optics. And this is the orthogonal orientation of this, and this is how the interface is relative to the chamber. From the top, you'll add your specimen. And this is called a stage up there because it drives at X, Y, Z and rotation. And this quickly goes to the imaging of live samples. I'll let it go. And so you can, with live samples, you have, again, perfusion CO2 that you can put in. The imaging chamber is made corrected for an refractive index of 1.33. For example, the agarose embedded zebrafish in a capillary. This capillary, again, goes into the top of the chamber. And then you push down the capillary. Your fish or your specimen arrives in front of the detection lens. This is how you position it, left and right, and you focus it and you rotate it. And so this is how you do the first positioning, and then you go to the light sheet imaging part, in which you simply say, do a Z-stack through this, and while the Z-stack is being done, you can see that this fish now translates actively via the stage through the standing light sheet. And it's, of course, ultra fast because the cameras work at this point in streaming mode, and you can do it within a few seconds. You can get an extremely large, like 1,000 Z-stack image. Cleared samples have their own chamber, meaning refractive index matching of the light sheet illumination optics of the glass of the chamber architecture. We just introduced a new smart sample holder that allows you to put the specimen directly into the chamber. There are challenges with extremely large samples, and so this is where kind of Damien and Translucence will come in a second, that do allow you to place whole brains, whole mouse brains into the chamber without trying to force them into a more diminutive chamber architecture. And again, this is the application. And now, for example, you can do this with tiling too. So you can have an XY translation of the stage while you do a Z-stack, and that allows you to, even at higher resolution, build a tiled image, like a full 3D tiled image of a large brain at the resolution you want. Then this directly translates through our built-in viewer and renderer. This is by Arivis. It's part of the Zen software and Zeiss into that 3D 4D image. The rest of this is just more highlighting kind of the technology. We are talking about the pivot scanner. There is different camera options on there. I won't bore you with this. And these are the specific chambers for cleared or living samples. And then there are specific objectives for these chambers and for these applications. So I leave it at this. I guess I do share my PowerPoint again. So this is kind of the first half of my talk, and in the second half, after Damien, I will then talk about a different method that is also a light sheet, but it's called the lattice light sheet, and go a little bit more into the technical details of how that works.
Okay, thanks, Oliver. So I think I'll just ask if there's people with questions for this first part, just type them into the chat so you don't forget them. And we'll let Damien share his screen and tell us about the large format sample holders. And then we'll circle back to Oliver for lattice light sheet.
Great.
So go ahead, Damien.
I have to unmute there. Can everyone hear me and see this slide on the screen?
looks good.
Perfect. Okay, so yeah, I'm going to tell you about one aspect of what we're doing at Translucence Biosystems that relates exactly to what was just presented in Oliver's nice presentation there. So at Translucence, we're creating tools and technologies to enable researchers, both in academia and in industry, to do next generation histology, 3D imaging of samples. We focus on brains, as many people do, because we're neuroscientists, but of course these methods can be applied in virtually any tissue or experimental paradigm you may work in. And we're developing things that will help with all aspects of these pipelines, of this pipeline for researchers. And we also do services within the company. So, but we have software for doing analysis, reagents, et cetera, for clearing and staining. But I'm going to talk about this thing here, which is what we call the mesoscale imaging system. And this allows one to do a little bit more with the Zeiss microscopes than was originally possible. So we were looking at the light sheet 7 there in Oliver's presentation, but when we started developing the mesoscale imaging system, the light sheet 7 didn't exist, and it was the light sheet Z1. Or Z1, as people have been saying, and I am Canadian, so I should say Z1. But the light sheet Z1 wasn't adapted for tissue clearing. It was created, as Oliver said, to be for developmental biologists to look at zebrafish and things like that. And the problem was the chambers didn't have the proper optics for high refractive index solutions, especially what we were using, which is for iDisco, which is a fairly high refractive index. And also the size of the chamber wasn't ideal for these larger tissues. So when we founded the company just over 3 years ago, we had access to a light sheet Z1 microscope at UC Irvine, where our company was founded by myself and a professor from UC Irvine, Sunil Gandhi, and his MD PhD student, Ricardo Azevedo, who actually helped develop the iDisco Plus method and ClearMap software for quantification when he was in Marc Tessier-Levine's lab. So when we started, we built what you see on the left. And you can't sell something like that, but it worked. And this was through collaboration with Zeiss and some 3D printed parts, some things we purchased, and it allowed us to get okay imaging with high refractive index solutions and get started on the Zeiss microscope. At the time, virtually everyone used the LaVision system, now Miltenyi, but the Zeiss microscope had much better optics. It was just hard to use. So we started using it with this, and then Zeiss came to us and said, hey, this is cool what you're doing. The fact that you're able to do these tissues, wouldn't you want to make a commercial version of this to sell to our customers? Because then more people can do this. And that's why we came up with what you see on the right, which is a much nicer looking version of the same thing that we were selling to the Z1 customers to help them do high refractive index large tissue imaging on the Zeiss microscope. And it's now sold through Zeiss as an add-on for new purchases of the light sheet 7, or for people like UBC that have light sheet Z1 and want to use these things. So this just shows, beside the state-of-the-art chambers, this is pre-light sheet 7. So now with the light sheet 7, there's new focusable objectives and new chambers from Zeiss that do allow for imaging cleared tissues. But at the beginning, that wasn't even possible. So our system was made to allow you to do cleared tissues, but now the added value of our system is to allow you to do 2.5x mesoscale imaging, which can't be done with the other chambers. So you'll notice that we have larger viewing windows here, that the chamber itself is larger to accommodate larger tissues. And you'll see this screw on the front here that allows for some adjustments. And if I show it this way, you can see better that this can move back and forth, and we have these marks on the side, but it allows you to use different refractive indices. So rather than focusing the objectives, you adjust the position of the chamber with this, with the software, but also with this screw here. And we do iDisco based methods in-house, but this works on shield samples and clarity samples. You can do any method you want. And in fact, we recently, before UBC purchases, we had one of Maywich's beautiful samples from using the LifeCanvas tech solution, which I'll say gives, works really, really well. And we imaged that here. I would show the images, but they weren't ideal because we weren't used to handling those tissues, and they're a little bit squishier than our gummy bears. Their gummy bears are a little bit softer, and we got a couple of air bubbles in there, so it doesn't look ideal. But certainly, this works beautifully for the shield samples as well. So, what do you get in the kit if one buys this? You of course get the chamber. We also made these sample posts, which look similar to the ones that Zeiss has made with the LightSheet 7 after we created this. I hope we inspired them in a way, but oftentimes people just use plastic syringes and things, but we have these sample posts that fit in and they're magnetic, and we have 3D printed holders. It says 3 of them, but we have 5 different ones for different tissues. And the design is open source, so the CADs are available. And if people want to adjust them for their method and tissue of interest, they can make their own adapted holders and print them themselves, or we can print them for them. And we like people to then share those designs with the community so other people can use them. And other than all the screws and screwdrivers and different things that come with it, we also have these safety collars that go around the objective. The reason for that is Zeiss builds their microscopes very carefully so that you don't break them. But our chamber would break the Zeiss microscope. You could smash this into your objective and break your objective, and that's no good. Zeiss doesn't like when we design things that break their microscope. So we created these collars that go around the objective, and the edge of this will abut up against the chamber before the objective touches it. That way you protect everything, and it's good to go. And it comes, of course, in this nice box that you can get with it. So I pointed out the 2.5x. So this is important, of course, that if you go to lower powered objectives, you can image much more quickly. And with the optics of the Zeiss microscope, you can still retain very good resolution that's good for most purposes. Sometimes if you want to look at the shape of microglia or something, you want the higher powered objectives, but the 2.5x works very well for most purposes where you're looking at cells and counting cells. And I'll just give you an example here. This is not interesting biologically. It's a Thy1 GFP mouse. With our methods, we use an anti-GFP antibody that soaks into the brain to stain the GFP. So this is not the endogenous fluorescence. And you can clearly see individual cells with this 2.5x resolution. You can see descending fibers here from the corticospinal tract. So you can get, this just gives you an example of the types of nice images you can get using the 2.5x imaging. I'm just going to go, I don't have much time, so I'm just going to go through a couple more examples here, and we'll leave it there. So this is a brain. This may be even the same brain that was on Oliver's opening slide. This was imaged on the LightSheet 7 at a workshop at Harvard just before the pandemic started, using the mesoscale imaging system for the 2.5x image there. This is work from our stuff at Translucence, where we're looking at amyloid plaques, and our software, machine learning enabled software, can go through the whole brain. This is an optical slice, but of course this is a whole brain image, and in 3D do analysis to identify individual plaques and map them onto the reference brain and provide you quantitative data. And you can do this. This is amyloid plaques. You can do this for virtually anything. This is work we've done with a immediate early genes. CFOS is the prototypical one, but we've got a proprietary antibody against NPAS4, which was shown by Mike Greenberg's lab to have a much better fidelity to response to neuronal activity. And we find that it's a better marker of neuronal activity. And you can see that in this paradigm where you keep the animals in the dark and expose them to light, you turn this on in the visual cortex, of course, but also in many other brain areas, including the anterior olfactory nucleus that you might not necessarily expect. And this just shows the kinds of quantitative data you can get out of your samples. using our, our software in-house, but it's also been made, a version of it is freely available, released by the Gandhi Lab, called BrainQuant 3D. I'm guessing, like LifeCanvas Tech, it's probably not, well, I don't know LifeCanvas, the text one, but ours is not easy to use, the free version. You need to know how to code Python and things like that. But we are creating user-friendly commercial versions of the software right now, so that can be useful, and we can do analysis as a service. This is just an example of some spinal cords. We have movies of this, but I didn't have it available. So this is part of a SARS-CoV-2 study where we're looking at neuroinflammation in the CNS and the spinal cord. So you can image spinal cords. And I'll leave with this final movie here, just because I find this fascinating. So Sunil, our co-founder, with his colleagues, Matt Blurton-Jones and Rob Spitali, they have a strong collaboration where they're looking at iPSC-derived human microglia, and they can use some tricks to get rid of the endogenous mouse microglia in a brain and then transplant into the mouse brain the human microglia. And it's amazing. What you see here is that these human microglia spread throughout the entire brain, and you can basically replace all of the microglia and make a humanized brain in the mouse with the microglia. So I find that pretty fascinating, and I think I'm probably used up all my time, if not a minute or two more, hopefully not too much, but I will stop there. Thanks.
Thank you, Damien. So again, I'm going to say, if you've got questions on your mind for Damien, just type them into the chat so we don't forget them. And I'll invite Oliver back to continue on to the lattice light sheet microscope.
Okay, I guess I'm back. Thanks, Damien. That was great. Awesome. Good stuff. I always love to look at these things because, when you see these movies, it represents, I don't know how many hundreds of thousands of hours of people actually working on it. And I think we always forget that that hard work. I mean, the translation of these, and so people who don't do this, and you guys very much understand this. But I feel when you look at this amazing image behind Damien is that this is a way that you can assemble this information that kind of, it almost honors our ability and the work that goes into a large picture, scale information. It's like the dream of the Google Earth of biology, like this dipping in and out and revealing these multiple layers of information. This is a cool thing about it. Anyway, not to get too philosophical about it, I will share my screen. Share screen, where am I and how does this work? Share screen. Back to this, share, and I will go slideshow from current slide. Okay, we were here, now we're gonna go to this. So another microscope that doesn't look like a microscope. Confusingly, it also has the name light sheet in its title, and it's called light sheet 7. So as for us as a company who make a commercial solution of these instruments, we now have 2 microscopes that don't look like microscope that are both called light sheet 7. Just the one has lattice in front of it, and it gets totally confusing. The idea about both of them is that they act as bringing in non-classical illumination in order to basically do a different modality of imaging. But this is kind of where it stops. Explaining the Gaussian light sheet, as I tried to do in the last the 15 minutes that I had, is relatively easy. Explaining the lattice light sheet is a little bit more complicated because it involves nonlinear optics. So before I go into too many details, well, actually, I would say I will not go into too many details because I can't, because of the time constraints, and also I'm not the right person for it. So I just want to mention that the lattice light sheet methodology was pioneered by Eric Betzig, and he has worked very, very hard with Zeiss in the last 10+ years to adapt his original kind of invention. into a kind of a benchtop microscope that can be used in any lab. The important part about the lattice light sheet is that it doesn't have the constraints of the Gaussian kind of waste beam, thicker, relatively thicker sectioning illumination, but it uses a nonlinear technique called bezel beams that are bunched together through optical engineering into a single lattice. And we use this single lattice to illuminate a stack of images at once. From the left-hand side here, you can already see that our illumination, here's one illumination objective, and here's the detection objective. They are at an orthogonal angle to one another, but not relatively to the specimen, which makes it more complicated. So let me jump through this. Again, why do we need light sheet microscopy? We can, what is it? It's basically the selective plane illumination. Again, this is a classic light sheet. It's a scanned Gaussian beam, and as well as it works, it is too thick for really revealing subcellular details, because the depth, if you match the depth of field or the depth of focus with the thickness of the light sheet, you end up with a multiple micrometer thick optical section. So a bezel beam or an array of bezel beams, when they are formed as a nonlinear technique, do have multiple side lobes or side beams. These side lobes you can see here with a thinner beam in the middle, which is called a scanned bezel beam. In order to create an optical lattice now, you have to coherently overlay these bezel beams into a lattice light sheet. And this lattice light sheet now allows such thin excitation light sheet creation that it can focus on the light in an extremely small area. Now, when this technique was developed by Eric, it really was developed for creating an illumination technique that is, and I don't like to use the word, but I always use it, as gentle as possible. It was really built around the ability to reveal or to apply continuous imaging of living cells over multiple minutes, hours, and even days. And there's now a lot of information out already, published information that shows, for example, here, continuous volumetric imaging of a Z stack. That's a stack, and this is basically, you always image a Z stack with a lattice light sheet over 9 hours. And you can see very intricate, fast movements, lamella-podia, filopodia, cytoskeletal rearrangement of the cell, and there's absolutely no indication of either photobleaching or photo damage in the cell over 9 hours. And this simply is not possible with any other technique. So how does it work? In a little bit more detail. I'm going to show you a beam an animation here. So there's a beam shaping optics in which is a laser beam gets shaped into a flat optical sheet, and then this sheet is being transferred into the into its spectral components, and then that bezel beam is built, and then the bezel beam is dithered into a lattice light sheet. Now the lattice light sheet is projected through your excitation objective, and you can see it interfaces with your sample through a meniscus lens, and this meniscus lens basically decouples your excitation and detection optics from your cover glass. And or you can also see it comes in at an angle, so it always excites a 45-degree angle Z stack at the same time. So what is being detected by your emission lens is now not cover glass aligned. So you can then throw different I guess emission filters in there and detect this lattice light sheet. So here's the setup. Here's kind of the waist of the light sheet, and just focus the waist of the light sheet throughout your whole kind of Z extent. So this now will represent the focal plane of your detection objective. You can see, since it takes a kind of a 45-degree angle section through it, is always what we capture always as a light sheet or as an image is a Z-stack image. And then the Z-stack, and we can actually see this in the focal plane when the dithering is off of the light sheet, you can actually see these Bessel beam patterns, or the lattice patterns. And by translating the stage now, the cell relatively to that fixed light sheet section, we create and are able to image an extremely fast Z-stack, continuous Z-stack through a cell or through a sample. So the sample scan is done by the stage. So in fact, it's actually, it moves in Y direction, so to speak. So the Z direction is built into the sampling volume, and the sample moves through Y. That means the raw data that we're going to get is a continuous a section through different parts of the cells moving in Y. And so this is basically what the camera sees on the right-hand side while your sample moves through Y. And this is the XZ camera, XY camera view and XZ camera view. And to then build a meaningful image out of this, we have to take this raw data that this camera sees and relatively translate it to a meaningful XY data set. This is called de-skewing. And you can see the de-skewed image here. So you can see it goes from kind of this elongated YZ. You can see this here to this data. And then there's another step now that while we now have this de-skewed data, but it's still basically a horizontal transformation of your data. A vertical transformation of your data relative to the horizontal plane of your cover glass. We now have to translate it one more time, and this is called the cover glass transformation. So we make basically out of this image data set this image data set, and that processing is done on the fly. So you will get a meaningful image out after your image with this, but on the raw data side, this is basically what happens. So you can see here the illumination objective and the detection objective, and you can see here the translation of both of the excitation and the detection optics through this meniscus lens. This is a special lens that is actually made by our semiconductor manufacturing, and it needs kind of a standard, I guess, variance tolerances in the nanometer range. So this is why we deal with the semiconductor EM manufacturing with this. So this is the meniscus lens. Our objectives are coupled via the meniscus lens is coupled via water to your sample cover slip, and that allows for extremely high numerical aperture imaging. But your objectives are not coupled, so we have an uncoupling of our detection objectives and illumination objectives from the cover slip, which means that you placing your sample on here onto this inverted microscope makes it very easy and does not have any constraints. It basically handles like a normal compound microscope. It shows you another example here. You see dividing cells over multiple hours, and you can see a cell that divided in the very, very beginning. I should kind of go back. I think it's kind of the first cell down here, splits up in 2 cells. And this is hours, 4 hours, 5 hours, 6 hours, 7 hours in. And you will actually see the same cell that already divided here, redividing again after 18 hours. Again, while this may not be a biological important or relevant thing, that shows you that there's so little phototoxicity that these cells, even after continuously imaging them for 18 hours, keep on dividing and don't photobleach. So again, the idea of this instrument is made in order to facilitate impact-free or impact-minimized lifestyle imaging, and that's really a totally new frontier for that. Having talked about hijacking before between lifestyle imaging, developmental biology, and deep tissue imaging and neuroscience, I'm going to show you this because this is an imaging stack through, and I have to look this back up, I think 100 something micrometers, also taken with the same microscope, the lattice light sheet. And this is a sample that was prepared by Anne-Marie Craig's lab and was sent to our demo labs in California. And a colleague of mine, Will Briley, I should go back here, so sorry. It does things that I don't want to do. I think I'd have automatic slide advancing on this thing. Took this image, and what he basically did is he took multiple built-in Z stacks and built a Y stack, a Z stack of these Y stacks, or a Y stack of these Z stacks, and was able to fuse the data and basically extended the applicability of this lattice light sheet, which is extremely fast, to a large cleared stack, which also allowed us to identify a single synaptic puncture. So here the ability is to resolve extremely fast, extremely large volumes, but with a resolution that's unmatched. So this is kind of in a nutshell what the lattice light sheet does. Again, I'm going to show you a little promo video because this will highlight how simple this is. This is filmed in real time. Kirsten, who's the product manager for this, just basically puts the cover glass in. No alignment necessary because, again, uncoupled with decoupled optics. There's a transmitted light that you use to focus, beam that you use to focus onto your cover glass, onto your cells on the cover glass. And once you're focused, all you have to do is start the experiment and image through that whole stack, and you can see how fast it is. So in terms of ease of use, this is extremely easy to use. This is a non-D-skewed image data right there. You can see that continuous Z stack or Y stack, and this is on the top the D-skewed, on the bottom the non-D-skewed, non-D-skewed and D-skewed version. So this makes it really into a functional kind of single button click microscope. It has a lot of stuff going on for it under the hood, of course. I will not go into too many details, but some of the stuff that you'll see is like full incubation, because again, this is built for live cell imaging, but it also has auto immersion. And in order to align this, alignment is absolutely crucial here as a fully built and motorized 5x tiltable stage. So 5-axis tiltable stage, and this stage can be auto aligned relatively to the light sheet with a button click in the software. Last thing to show you is a couple of examples, and I guess, you know, Jeff asked me or suggested to show you some images and figure out whether these images were taken with a Gaussian light sheet or lattice light sheet. So you can take your guess here, but I think you possibly know. So this shows on the left-hand side flower development or petal development in Arivis plant. There's a fully rendered brain volume that you see here, and then there's some Drosophila development on the right-hand side. So this is one set, and then the second set is this data set. Oops, I should start this here. It's a different data set. Again, showing a volume. You can see this here when it tilts over a relatively large, long time interval. So if people have a vote or want to think about which images could be taken with which instrument, you can be my guest. And there's lots of other images to share, but if there's any more questions about the comparability or compatibility between these two techniques, please let me know. That's it from my side.
Okay, welcome back, everyone. It's a quick break. So we're into our final session of the workshop. Our final session is going to discuss data analysis, and we're pleased to have Maria and Molly here from Arivis. We began looking into Arivis when we were writing our proposal for the lattice light sheet microscope, as it is a software package that is capable of handling the kind of enormous data sets that come from this type of microscopy. And we've been working with Molly and Maria to set up pipelines that will help us analyze the data we're anticipating collecting with such a microscope. So we'll start with a presentation from Molly just to give an overview of the capabilities of this package before we move into a specific example and demo of a pipeline that we've been working on with Maria. So Molly, if you're there, could you share your screen?
Yes, I can. And then you can see that.
Yep.
You should be able to see this.
Yep, we see it.
Perfect. So thank you, Jeff. As Jeff said, we're going to be focusing this last talk of the workshop on how to handle these large data sets post-acquisition. And we're specifically going to be talking about the Arivis scalable imaging environment. And more specifically, we're going to focus on Vision 4D, as Jeff mentioned. So to start off, we're going to touch on a couple different challenges probably many of you are familiar with as imaging scientists out there in the field, and really how we got into the market and what we have to offer. So the first is that the tools for working with big data, such as that data made by these different systems, these systems you just heard in the last talk, are severely limited. And what do I mean by that? In order to efficiently work with data without the pain of the software being slow, being laggy, waiting for the data to process just to know if the process will work, the data can only be of a certain size. And that doesn't always work for scientists depending on what your research question is. The second is there's a multiple of disconnected tools for pre and post processing in the market. And this is great because it shows in the community there are a lot of people trying to solve different imaging problems and offering tools, but it also hurts anyone that wants to leverage these tools in a research or production environment. This is because a lot of the time, and probably more time than anyone realizes, is needed to integrate and leverage the available tools in a productive way. Lastly, and what we're going to touch on very briefly at the end of my talk, is insufficient throughput. Every one of you has to look at lots and lots of data sets, process lots of data sets, and this comes with a whole host of challenges. So increasing throughput is not easy to do, but it is necessary in modern science. And we, as members of Arivis, care about these challenges. And what we've done as a company is tried to offer solutions to help scientists with each of these challenges. So we're going to start with the first one, and probably the most important today, which is working with tools to handle these really large data sets. So as you maybe already knew, or you've definitely learned in the course or the workshop so far, is that more techniques have come out and they have led to a severely increased data size for individual acquisitions. And this is not only an increase in XYZ dimensions, but it's also in time and channels. And the issue which arose from this acceleration in data production is that the hardware has not been able to catch up, resulting in a gap between the need and the hardware available for processing. So that's one issue of this challenge. The second is data production is also parallelized, meaning you are collecting more than one image at a time. You're collecting full experiments worth of images, and you're likely running multiple experiments at the same time and leveraging multiple microscopes. So we not only have a data size issue, but also a data quantity challenge. And our solution to this and solving this challenge of working with big data in an effective way is how we really came into the imaging science space. It was with our first solution, which is our SIS format, or our scalable image storage file format, and our image core technology. These two tools are the basis of all of our software products, including Vision 4D, which you're going to see in a live demo today. So what is the SIS format and the image core technology, and how do they work? The SIS file format is a proprietary file format of ours, which allows files to essentially be limitless in size. So you don't only have to store a single massive data set in one SIS file, you can also store your entire experiment worth of files within a single SIS file. Our image core technology is our way of interacting with the SIS file, and it scales with the hardware. So one way you can think about this is a smart layer between the huge amount of data in your sys file on the disk and the available computing resource. Whether you're working on a laptop or a workstation for analysis in the core, whatever, it's the image core technology sensitive to this and uses all the available processing power appropriately. As a result, you get full interactivity and stability at any zoom level of the data. And this allows you to have very fast feedback, easy handling of the images, and smooth interactions, regardless of what size that sys file is. Once we came out into the market with the sys file format and the image core technology, we realized we could help in another area, and that is that there is this multitude of disconnected tools for pre and post processing. And this is where we're going to focus most of the talk and where the demo is going to focus, because our solution here is Vision 4D, which you'll see in a moment. So what is the challenge here? The challenge is that all of you want to acquire your data on the systems and then get to your results. And you want this to be in as smooth and simple and efficient of a process as possible. However, this is in reality not normally the case. What we see is most times people have to start in one solution, let's say open source for the sake of the example, to do some portion of their post-acquisition processing, then move into another software in a different environment. Maybe it's a commercial solution available to you. Maybe it's custom code, something else. Do a portion of their analysis, move into another environment. And often this process even cycles back multiple times before you can get to your results. And it ends up being very laborious and time intensive. What we put on the market is Vision 4D. It has the back end of our large data handling with our sys file format and our image core technology, but it also is a fully integrated toolkit. It is built on 4 principles. Flexibility, so you can address any research question regardless of the imaging modality. Accessibility, so that a novice or an expert imaging scientist can use it and analyze their data. Extendability, so that an image analysis expert can leverage all of the tools needed at hand for the task. And scalability, so it's applicable to a single image or an entire experiment worth of images. As I said, it's a flexible solution and a flexible toolkit, and it's designed that regardless of if your research question is cancer biology, cell biology, neuroscience, plant biology, anything, you can use Vision 4D as a solution for your image analysis needs. It is also built to be image analysis or image modality agnostic. So whether you're working with light data, particularly light sheet data, or confocal, maybe you're working with EM data or even medical imaging, for your research question at hand, you can import these images into Vision 4D and leverage the full toolkit for your needs. We wanted this to be as applicable as possible to help address this challenge. So with this toolkit, Vision 4D allows you to leverage a variety of modules and operators and different tools to go in a relatively straight line from your acquisition to your results, particularly in a way that you can stay in one environment. So you can stay in the Arivis environment. What you'll hear in a minute is that even includes connections to open source and custom coding. So if you do have to extend the software, we try and make it in a way that you can still stay within the environment and not move your data around, which if anyone here has worked with these images firsthand, which I know some of you have, from systems like the Lattice, that is a challenge to move these large data sets in between different environments. So being able to stay in one is really a benefit. Okay, so I'm going to highlight and give you a sense of a lot of different tools in Vision 4D. This is not an all-encompassing list. So if you have questions about a specific operator, tool, or algorithm, or anything related to your question, please pop it in the chat. I'm happy to address it. But to give you a general overview of the different topics or the different tools Vision 4D has, we're going to touch on some main ones. So a big one for specifically for light sheet imaging and clarity is stitching. We have 2 types of stitching tools. We have XYZ stitching and we have volume stitching. You're not going to see these in the demo, but we do have them available. And of course, we'd be happy to show anyone who's interested at another time. We have a lot of visualization tools. We have multiple rendering options and a whole new object style module, both of which you'll see some of in Maria's demo. What you won't see is that we do have a full animation tool and snapshot tool for outputting your data for presentations. So this is where you're going to make cinematic renderings, some of which maybe you've seen throughout the workshop, and high resolution snapshots for your papers and your presentations. We have a very dense list of segmentation operators. To highlight 3 that you're not going to see today, we have seeded region growing, a very commonly used one called blob finder, which is very good at finding round objects in noisy data. We have membrane-based segmentation and a ton of others. You are going to see a lot of intensity threshold segmenting today in the live demo, but if you do have any questions about particular strategies for analysis, just let us know in the chat and we can answer that. We have 2 types of machine learning, pixel-based classification and object-based classification, both with very easy to use user interfaces. And an area where the demo will focus very heavily is on our object relationships. You're going to see a lot about distances and hierarchical analyses of objects. What you won't see is that we do have a full tracking module. So if you do have time series data, we have a full module dedicated with tools for analyzing this type of data. As I said in the last slide, we also have a full Python script interface in Vision 4D itself. So you do not have to leave the environment to leverage this for your needs in case any of our traditional methods do not work for your research question at hand. We also have a connection in Vision 4D to open source so that you can bring things in and take things out if you really need to in a hopefully painless way. So one major benefit of the flexibility of Vision 4D and all of these operators that are available is that it allows image analysis experts to leverage these for segmenting very, very complex objects. Many of you here probably know that when you are working with neurons and you are working with membrane stains of neurons, they're often broken up, they have holes. It is extremely challenging to easily segment individual neurons from these type of stains. Through a combination of the tools available in Vision 4D, you are actually able to get segmentations of these types of challenging objects in our system. As you can see here in this bundle of neurons, you do end up with segmenting these 3 individual neurons present. So the last point before the demo that I really want to make about Vision 4D is that it is inherently scalable. We do have a batch analysis module that allows you to apply your analysis pipelines to not only a single image, but sequentially to an entire experiment worth of images. This inherent scalability is what brings us to our last challenge, which we're going to touch on briefly, which is increasing throughput. I touched on this in the beginning, and it is very challenging for any of you maybe that have this problem right now or foresee it in the future to scale analyses in a production manner and actually run analysis in parallel. Yes, I said Vision 4D is batchable, but it's in a sequential manner. It cannot itself parallelize tasks. It is, however, compatible with another Arivis tool built specifically for this, which is called the Vision Hub. And I only want to bring this up in this talk today because especially as you're working with these larger data sets, this does become a problem and a need, and it's something you should keep in mind that we do have a solution for this. So how does the Vision Hub work? So it allows multiple users to be working in Vision 4D, building pipelines for their analyses with their images, vetting them, getting them ready, and once they're ready, they can send these analysis jobs to the Vision Hub. And what the Vision Hub is going to do is it's going to take these jobs as they come in and allow them to use available computational capacity that's registered to the Vision Hub. And it's going to do this in a scheduling manner. So if all the computational capacity is used by these 3 users, this user, he'll schedule his jobs, it waits, and then as soon as some computational capacity opens up, those jobs will start running through the Vision Hub and be applied to the new free computational capacity available. What it allows you to do is, as I said before, parallelize this process in a way that you can get from your images to your results much faster than in a traditional sequential batch process. Okay, so what I want you to keep in mind from this portion of the talk as you go into the demo from Maria is that Vision 4D and the Arivis products have an environment which is smart handling of multidimensional data sets. It is a flexible tool set to analyze images regardless of research topic or imaging modality. It's accessible to either the novice or the expert. It's extendable via scripting and open source in case the traditional methods in here are not enough for your research question, and it's inherently scalable and in connection with other Arivis solutions. With that, I believe Matthias is going to be giving a little introduction into the research question for the data being used at the demo, and then it'll be passed off to Maria, and of course, then we'll take any questions.
Yes, so I'm just gonna share my screen.
I will try and stop sharing.
Okay, so I think now you are seeing my screen now.
Yes.
Yes, we are.
Okay, so very quickly, what Maria is going to show you, what kind of data Maria is going to use for the pipeline is my data. So we are interested in the synapses of expanded tissue. So for that, we are staining synaptic proteins. Here, the synaptic proteins that we are going to look is in red PSD95, a postsynaptic density proteins, and in green, it's going to be RIM. So as you will see, what we want to do is to localize this puncta, so the green puncta and the red puncta, and do different kind of analysis. So for example, the distance between this puncta, but mainly localize this signal and do a bunch of analysis on this signal. So using the expansion microscopy, we can see clearly these synaptic proteins. We can identify them and separate them, every synapse individually, as you can see by the alignment of RIM and of PSD95. You can see the different synapses. So this is what we are going to study during the pipeline. And so why we are going to Arivis is because of the future data that we are going to have with the latest light sheet. As Oliver mentioned earlier, the data that we can obtain with the latest light sheet are very big. It's huge. So as you can see the dimension here, in X, we are almost at 300 micron. In Y, we are at 130, and in Z, we are at 16 micron. So with the software I was using before, I couldn't manage this kind of huge data, but with Arivis, it seems to be able to handle this kind of huge data. And so I'm just now going to let Maria take the relay to show the pipeline that we worked on about this analysis of this data.
Yeah, thank you, Matthias and Molly, for the introduction both to Arivis and to the biological problem we'll be looking at. So I'm sharing my screen, and let me know if you can see it.
Yes, we can see it.
Yeah, okay. So basically, we will talk about how to get from the raw intensity values to fully segmented particles for PSD95 and RIM. So here we have the Vision 4D software. Maybe I make this a bit lower so that everybody can see absolutely everything. And yes, so we have two channels here, the green and the red, and also we have the light sheet channel. And we already have the particles overlaid, and right now I'm showing it in the 4D viewer, so in the fully rendered mode. And I can rotate the whole stack the way I would like to. And of course, we can zoom in much closer on individual particles and access them. We can also interact with each of the channels. We can change the opacity settings from here. And right now in this image, we are having only in the navigator, in this SIS file, we are working only with one image stack, but as Molly mentioned earlier, there could be more than one, and the basically contain the whole experiment. So I will change now to 2D viewer and show you how the data looks like, so we can also disable visualization of all the objects. So I was in the top slide, and now you see how we move through all the planes. So this was acquired with the lattice slide sheet. So you can also appreciate that actually because of this queuing and assembly of the final data, it also has black stripes on the bottom of the stack that we can visualize from here. So analysis is probably one area you're most interested in. So we have the whole panel on the right, which right now contains already one pipeline that we will go through today. But just by clicking here, we can create a new pipeline, and it always starts from importing the ROI, and then we can add many operations. And in Vision 4D, they are grouped into 5 main classes, always starting from image processing, so from the operations that work on voxel to voxel, pixel to pixel based, then later followed by the segmentation, which means that we create an object via one of the plenty different methods that will be stored in the objects table that later can be again classified, or today we will use the distances operator to measure the distances and actually assemble the groups of the PSD and rim particles. And then once we are happy with our analysis, we can export it, and a separate group of operators is related to the groups and the tracking. The basic rule we have in Vision 4D is that we first always use the pixel to pixel operators, then later followed by segmentation, tracking, and export. And you can mix and match them within these groups the way you like, but not in between the different groups, which are also color coded here. So if I go back to home, I will open the pipeline that I have prepared for. We have prepared together actually with Matthias before. And first step is importing the ROI, where our whole stack is actually 2000 pixels in length. And for the sake of the demo and the time, I'm only taking a small crop, so 500 by 500 pixels in the middle of the whole stack, which is in this case 100 Z slices. You can also access it, the same information from the information panel on the left, right, if you need to. And here are the most important buttons, which are to move forward with the pipeline, or to execute the whole pipeline, or move backwards. So now we will be focusing on the smaller ROI on the right left upper corner, and immediately we can go to the intensity threshold for the red particles here, and this is the preview button, which would visualize all the particles that would be identified with the given intensity threshold. So why we are using intensity threshold in this example is because we can also change their opacity, is because the shape of the particles is often non-round, and even though they would be perfectly identified with the blob finder as well, their shape might be distorted. And because we want to measure the distances and the volumes of those particles, it's important to preserve their shape as well. So I will execute the next step of the pipeline to segment some particles. And here we have the objects table, which can be just a separate pop-up window, which now is populated with the all PSD particles that we can visualize here in yellow, of different sizes. And now we have to identify also the segment the particles for PSD 9 for the green channel for the rim. Here they are. Again, it's a simple threshold, which means that all the intensity values above certain level, which in this case is 237, will be accepted as the particles, and we will create the objects from there. So I will run this step as well, and I can fold up the previous steps. So now we have both of those particles, and many of them overlap, and but you see that we also have several small ones. So especially if we visualize this in 3D, we will only have a small portion of the ROI here that we want to focus on. And yeah, you see many of the particles, especially if I remove the channels, are colocalizing, but also there are many of the small ones. And in anticipation of one of the common questions is that all the operators are performing in 3D if you tell them to do so. Now, next, I would like to filter the particles by their size, because some of them are clearly too small and some of them are clearly an aggregate of something. So we can combine that in one operator here, where I'm just saying that for the particles for the PSD channel, please take all the particles which are larger than 100 voxels and smaller than 2,500, and I execute it for one channel, and it's exactly the same step for the rim particles that I'm using here. And now, again, in the table, we will have the filtered particles only, as opposed to the raw ones. So now that we are happy with the size of the particles that we include in the analysis, we can actually run the step which will create the groups. So basically, this is the hierarchical operator to group together the particles of PSD and V-glued that fall within a certain distance from each other. Again, it's easier to visualize without the channels. So we have PSD particles as references because we know these are the yellow ones in this example, because we know that typically there will be one particle of PSD 95 per synapse for postsynaptic density protein, while there might be more than one particle for rim staining. And from the previous experience, Matthias suggested that we should only include particles within 600 nanometers from the center of geometry of each yellow particle. And here we create the groups, and we don't limit to the closest, meaning that we assume that there will be more than one rim particle per single PSD particle. And with this, we will also store the distances in the objects table on the right, and we can also export this as distance matrix for each, quantifying the distance between each of the two particles. And also here is the name of the output that I actually did not introduce today yet, which is basically we'll give a tag name in the objects table. So here are the tags for all the objects we have so far that we can later access separately if we and distinguish this group of particles from other steps of the analysis that we are doing. So I will run the distances operator, and let me switch to the viewer. Now we should have a new group here, which contains 153 groups. Each group is representing a synapse, which consists of one PSD particle and several of the rim particles. And here, if I change to master detail mode of viewing the objects table, we have we can access and click for every synapse and see that it consists of one PSD particle and one rim particle in most of the cases. But now if I align it by the number of children, on the whole stack, I had several examples where there was more than one example of rim particles there. And with this, I would like to actually show you that we are also able to bin the particles by the distance. And what I mean by that is that now I am running a very similar operator, but I am, instead of taking the closest particles, I'm actually taking a range from the distance we took before and doubling it, meaning that we will have two groups with different names for the analysis. And how I can do this is using the filter operator, where we have, of course, like more standard operators, such as taking the particles which are closer or shorter than certain distance, or taking a range, or taking accepting the particles that are outside of the certain range, but would be positioned elsewhere outside of the distance between A and B. And now I run this. And we will have it stored in the objects table as well. So right now we're in the analysis mode, where this is basically access to all the segments that we are working with, but we also will store them in the very last step of the pipeline, which is always there by default. And before doing that, we can also export the existing particles into the Excel or CSV tables. And what I wanted to show you before is that while we're running the analysis, we can, for example, here also quickly visualize the quality of our analysis of the segmentation by, for example, plotting the intensities to the volume values for each of the particle types, even simultaneously. So here the cutoff was higher because we only accepted the particles which are starting from 50 micrometers of voxels compared to the rim particles. And yes, so for the export here, we can use this very similar master detailed report that exists as one of the options, and populate the selection with the same features that will be also exported, that we can visualize and compute live for the particles. So in this example, we go to the master detail report. We have all the children. Also wanted to exemplify here that some of them actually will have more than one child object per PSD particle. And for each of the particles, we can also export the distances. So this is the distance between the PSD particle, which is the top one here, and each of the rim particles between the centers of their geometries, exactly as we have indicated here in the operator. Because in some cases, we have more than one rim particle per synapse, and because we actually create the groups for each of the synapses, which a group is basically a new type of object that exists in the Vision 4D. Was one of the biological questions, for example, to quantify the ratio between the intensities in the green objects channel compared to the red ones, or the volumes of those particles that we have segmented. We need to create custom feature which would represent the group statistics. And I already did this for volume and for the intensity. So as for each of the columns. And let me show you how to do this. So if the column is not populated, it means that the data is not accessible for this kind of computation. But let's create a new one. So we go to the custom features, and we will create a new group statistics feature, which will have a name, and we will quantify, in this example, the volume in micrometers between for all the rim filtered particles, as in this example, and we will take the sum because we know that there's often more than one in the analysis. And when I finish this, I can close the table, and I now can access the feature that I have just created. And here we have the sum of, in example with 3, of all of both rim particles together for the analysis, if it has to be. This is just an example how we can further customize the software with the and compute new features that if we have to. The same way, once we have the sum of each of the particles, we can also create the ratio and immediately populate the Excel or CSV table with it. So to export the features, we can basically give the path and the file name, and run the step, in this case both for each of the 2 groups of particles. And as the last step, I'm basically saving these particles again into the objects table that can be accessed. It is already stored, and it can be also exported if necessary. And now I actually, because I ran this pipeline before, I actually have it twice for all excuse me, all the particles from earlier for the whole stack. And actually, as Matthias mentioned, the file is pretty large, but on this same pipeline in my computer took less than 5 minutes, I believe, to run it. So it's still pretty pretty small actually. Now, what I wanted to show is that at the end we can have the same table accessible, and what we can do here, for example, is to again, we have the names and the IDs for every of the object, and if we were to compare the particles in either of the groups, in the small group from the closest to 600 objects, nanometers, to the longer one, we would be able to do it using their tag names. And what else we can do is, for example, access quickly the aggregated, in this example, intensities for rim particles compared, plotted against how many there were per volume, per synapse. And yeah, this basically brings me to the end. I can I saw already one question from Mavish about the vesicle tracking that I could address immediately as we are talking. So the question was about vasculature and whether we can do the tracing. And yes, we have working on this from 2 fronts. From one side, we have developers team working on the long-term solution to create a new module, but right now we actually just very recently we had a new script developed by one of our colleagues, which script does is it creates a skeletonization of all the vasculatures, so they would look like 1 voxel thick lines that later can be computed. And what we have there is that the script divides this skeleton structure into individual branches for which we can quantify the generation number, their length, their like real and linear length, so that we can do the tortuosity measure as well. I hope that answers the first question. And yeah, I will switch back to excuse me, to Zoom so that I could see the questions. And I guess I have to stop the screen share for that.
The second question, Maria, was in regards to if we have a support portal or any sort of source for the community for scripts and pipelines, and I provided the link to the support portal. The second question left was about importing Allen Brain Atlas regions into Vision 4D and doing the scaling.
Yes, so we can doing the scaling or
yeah, so how
like matching and rendering. Okay, or
yeah, so what I wrote in the chat, but we can probably expand on a little because I know you've worked on this a little bit more recently, is that the elastic transformation is being done in other software. Specifically, a lot of our customers are using ClearMap to do the registration.
Yes,
but they're then bringing them in via label TIFF images into Vision 4D to do the downstream analysis.
Yes, so what we can do from there, how we can take over is that once we have brain regions annotated, and for example, the cell density is calculated in those areas or some other parameters which are of biological interest, we can take this usually a CSV or TXT table and match it to the model brain, so to the Allen brain, with to the model based on the labeled image segmenter. So the what labeled image is means that each of the objects will have unique color for all those pixels which belong to this object that can later be used as an ID to match the between different brains. And once we have the ID, we of course can also import other features and values associated with it, such as cell density, as I mentioned, or even the behavioral data for before the brain was fixed, as an example.
Maybe Damien, you can go ahead with your question, and then we'll come back to Honor's question in the chat.
Sure. So I think you partially answered it there, but my question was about you used a sort of hard cutoff threshold for identifying the objects, in this case, synapses. And it was clear that there were synapses in the image that were not detected because they were below threshold, but if you change those thresholds, you're going to get false positives, etc. So you mentioned, or Molly did in the intro, about having machine learning algorithms, and you must have a bunch of other filtering processes, etc. What can you do within the software to be smarter about identifying the objects in the very first step? Or is the solution using Elastic or something to train outside and then bringing in a probability mask into your software?
Yes, so first of all, of course, we can use adaptive thresholds in the pipeline that would or just set the threshold at the lower levels. Once we have that, we can, so this is the second part of the answer, is that once we have many particles, including the potentially false negative ones, we can go to the object machine learning tool and classify the particles from the objects table, for example, all of the rim particles based on their, it could be feature, their intensities and morphology, but also we can create the custom features that will basically distinguish the existing objects into the real ones versus the dirt, the leftovers, that can be later taken into the analysis. This is done pretty easily. So what I need to do is I need to select my one type of particles. Let's take, yeah, we have only the filtered ones, and for all for the whole stack. And in this example, I can just say that this is probably too small, and this seems to be the correct one. And once you give about 10 images for each of them, that would likely be sufficient for the training. And already after 2 examples, we can here preview the result that it separates the particles by size. And now that if I switch this back on, I can say that this is a correct one as well. And so on, very quickly, we can get a pretty good classification. That's the second part of the answer. And the first one, yes, is that once especially if the data is more complex than this, we don't have to go to Elastic. We also have the pixel-based machine learning, where we can use information from one or more than one channels that are available and paint the objects of interest, let's say this, and maybe this. And we also need to, of course, give the background information, then later can be trained. And we can also, predict the results. So this is a very early for prediction. We would need to paint a bit more, but you have a taste how this works.
Cool. Okay. Yeah. And do you have other filtering things in there, like rolling ball backgrounds or Gaussian filtering, etc.?
Yes.
Pre-process the images.
Oh, absolutely. So all our filter operations are in the orange group. For example, we can start from background correction, which would subtract either a blurred image or a certain constant, then normalization per stack or per plane or per time series, a certain part of time series. And for the denoising that you asked specifically, we have a whole bunch of filters here, including the Gaussian and particle enhancement, also very useful sometimes. It's not basically a denoising operator anymore, but rather to emphasize certain structures. And we have many more.
Nice. Okay.
Including morphology filters, and we can also create a machine learning probability that you have mentioned, which basically creates a grayscale probability map for a particle to be there. And in certain situations, you just use this and then threshold it for the following steps.
Yeah, that's great. Then and with your pixel machine learning and the probability mass, etc., are the algorithm that gets generated by that, is it hidden in the software, or is it something that can be extracted, used in a third-party application, and then bring data back in for visualization or something like that?
The algorithm behind both pixel and object machine learning is random forest, which is the same that is used in Elastic, for example. And it creates, right now we can customize the features that it takes into into the analysis. So in this case, that would be, again, in the custom version, we would have a certain size and 4 groups of features that we can use. Also, it can be downscaled. Once the objects are exported, we can export the objects, we can calculate a lot of values in them, we can create like a labeled image or a mask. But I remember that regarding the export, we can export the training as a Zeiss proprietary file format, but we can import it from the those trainings which were created in Elastic up from a certain version of the Elastic software. So we can bring them in.
Just to add on to that, so the actual forest output, which I think you're trying to get at. We don't release at the moment, but that's on the roadmap to be made as an output.
I see.
Yeah, we just haven't gotten that far for integrating it in the UI.
Okay.
And on the to-do list for the Arivis team, there is also the regarding the machine learning is to have a visualization or the output, which of the features that we're using actually give the most prominent result, so that you can prune the trees and the future algorithms more efficiently.
And really, sort of to follow up with one more, but we're doing it for the sake of the demo. You said you're doing a relatively small area here and you're looking at fine structures like synapses. But if I were to want to use this to identify a certain cell type in a whole brain image that the whole stack, can you handle images that size to create these probability outputs, etc.?
Yes, so we have for both of the machine learning algorithms, we have GPU acceleration machine learning, which is enabled here. So in the last release, which was 3.5.0, we already boosted the algorithm so that the computation is done faster compared to the earlier versions. And it's possible to run machine learning training and prediction for the both for the pixels and for the objects on 100 gigabytes and further scale.
Okay.
Yes, for the objects that have to be created before, we have tested it on 4 million objects and it runs.
Cool.
It is not very fast, but another advantage here is that I showed you that we can create custom features that can also be part of the training for the object classification.
Cool. Thanks.
You're very welcome.
So I just wanted to see if there's any more questions from those still on the call. I think Molly has answered Honor's question in the chat. Any final questions for Molly and Maria? No? Okay, then let's thank them for their talk. Thank you both. Thank you for the pipeline. Thank you for all your help while we've been working through this.
Very welcome.
Yep.
And thank you for participating in the workshop. And so I'll just share my screen to close us out then. So I'll just come back to this. I want to thank everyone who participated in this. This was really great. It's really great to have so many people coming together and joining us at UBC to talk about these things that we're all trying to get done in our research projects. I want to thank everyone that helped me organize this. Matthias, Sarah, Mawish, Nicole, and Lisa. And I think Nicole can paste the links in the chat again, but if you have follow-up questions, things you're thinking about, or you go back to the lab and you're trying some of these things and you're getting stuck, we have a weekly Zoom meeting every Friday afternoon. You can join and we'll try to connect you with the people that might be working on something similar and try to help you move things along. So that is the end of our workshop. Thank you everyone for making this 2021 version very successful, despite the fact that we couldn't do it in person, but maybe next time.